Hello, my name is Benjamin Butler, and I am a junior chemistry major at Nazareth College.
My name is Samantha Druce, and I'm a junior biology major.
And I'm Olivia Guerrette, and I'm a sophomore biomedical science major.
We are all undergraduate scientific research students under Dr. Charlebois within the chemistry department at Nazareth College. We'd like to thank you today for coming to our presentation titled Creating a Potential Biomarker for Autism through the Deuterated Reduction of Bilirubin to Stercobilin. A little bit of background info on stercobilin. So stercobilin is actually a biologically natural substance that occurs within the human body. It has also been found to be in a lesser concentration in the feces and urine of individuals that have autism spectrum disorder, also known as ASD. Stercobilin can also be artificially synthesized within a laboratory setting via the hydrogenation of another compound that occurs within the human body known as bilirubin. We hypothesized that being able to develop a deuterium-labeled isotopomer of stercobilin could potentially lead to the labeling of stercobilin as the very first biomarker for ASD. However, as we've conducted much research on this, we've actually found that classic mechanisms for hydrogenation do not work in the case of the synthesis of stercobilin, meaning this must be investigated further if we're to be successful in our research.
So like Ben was saying, we create stercobilin from bilirubin. And if you look at figure 1 to your right, the very top molecule is what bilirubin looks like. And as you can see, there are about 6 double bonds within that molecule. And then the very last molecule in that figure, which is labeled number 3, that is what we get, that is what stercobilin is. So how that happens is we use a process called hydrogenation, and we reduce those double bonds in bilirubin in order to get single bonds in stercobilin. So if you look to the lower left, in example 1 is the reduction of an alkene, which is a double bond going to a single bond with hydrogen gas palladium, and we used glacial acetic acid in order to get the single bond. And it's the gas that we use in the reaction that adds the molecules in order to get a single bond. So what we have done this semester and in semesters past is we've done a series of reactions to try and create stercobilin and try and mark it. So a basic reaction that we do is H2H, that is hydrogen gas and regular glacial acetic acid, and that gives us 12 hydrogen atoms added to the molecule. We do a D2D reaction, which is deuterium gas and deuterated glacial acetic acid, and that gives us 12 to 13 deuterium atoms. Now, like Ben was saying earlier, the mechanism isn't exactly how we thought it is, and the D2H reaction and the H2D reaction proves that. So if you look at the D2H reaction, the deuterium gas in that is what is supposed to provide the deuterium atoms to the stercobilin molecule, and we only get 3 to 4. And in H2D, we get 4 to 5 deuterium atoms, which that isn't supposed to happen because we didn't use deuterium gas. We also used, we also did an N2H reaction, which is a reaction we did to see what would happen if we didn't use deuterium or hydrogen as our gas. And we also used an H2THF reaction, where we use hydrogen gas and THF because THF doesn't have any acidic hydrogens that would react. And then this semester specifically, we used limonene and alpha-phalandrine to see what would happen if we took smaller molecules that looked like bilirubin to see if we can hydrogenate those double bonds within those molecules to see what would happen.
So a little bit of background on the data we use. The 3 main types of data we use are infrared spectroscopy, nuclear magnetic resonance, and mass spectroscopy. A little bit of infrared spectroscopy. So in IR, what we do is we would take a sample and we would see how infrared radiation interacts with the sample, which leads us to be able to determine what functional groups are within the compound. So then NMR, it is also used to determine what functional groups are within the molecule, but it does this by using magnetic fields. Mass spectroscopy, on the other hand, actually allows us to determine the mass of what compound we made. So in IR and NMR, we're able to tell if the reaction was successful, but in mass spectroscopy, also known as MS, that is a clear indicator of what was added to our sample, which is very important for this as we are determining whether hydrogen or deuterium was added. Hence, deuterium weighs more than hydrogen.
So like Ben was saying, this is an example of NMR that we get from our sample. So figure 2 shows a proton NMR of bilirubin, and figure 3 shows a proton NMR of stercobilin. As you can see with the different colored circles, there are different indications in bilirubin and stercobilin that we were able to get stercobilin and get those products, and that is how we know what our sample is like. So another example, like Ben was saying, is mass spec, and this is how we read mass spec. So in a sample of stercobilin, that gives us a peak of 595 because that's its molecular weight. So what mass spec does is it bombards a sample with energy and breaks it up, so we are better able to look at what the molecule actually looks like. So for example, like I said, this is a molecule of stercobilin. So a 470 peak that we see is this molecule, but with one of the chiral molecules broken. So at one of the red marks on the screen, one of the chiral molecules will come off. And then a peak of 345 would be 2 chiral molecules, which is both ends of the molecule coming off that we'd see on the spectrum. So this is an H2H spectra. So as you can see, we have our 595 peak, and off to the right, that is the structure of stercobilin. And then if we bombard that with more energy, we get this. So as you can see here, we have our 595 peak. We also have our 470 peak, which is our molecule with 1 chiral missing. And then we have our 345 peak, which is just basically the center of the molecule with both chirals missing. So for a D2H reaction, it's a little different. We get a 597 peak. And if we bombard that with more energy, we see this. So we see our 597 peak, and then, and that is 2 more than our regular 595 peak, which tells us that there are 2 deuterium atoms somewhere on this molecule. So then our 471 peak is 1 higher than our regular 470 peak we'd see with regular stercobilin. And this means that there is 1 molecule of hydrogen somewhere on the atom that is not broken. So reminder, our 470 peak is a molecule of stercobilin with 1 chiral missing. So that is what this is. And the 471 tells us that there is 1 molecule on the 1 molecule of deuterium on the rest of the molecule, and then there's also 1 deuterium atom on the chiral that we took away that is broken. And then if we look at our 345 peak, we see that that's a regular peak we'd see with an H2H reaction. So that means with both chiral molecules missing, the center of the molecule doesn't have any deuterium on it. So then we know that there is 1 molecule of deuterium on each chiral missing.
So a little bit on the D2H we have actually previously ran this experiment before, and this is in spring 2019. I'm not sure if you can see it on my screen. You cannot, as this is a recorded Zoom meeting. However, the highest peak is indicated at 601.386, which, as Sam was just saying, with the 597, this value is actually much higher than that. And this actually is 6 higher than a normal stercobilin, allowing us to believe that only half of the reaction was successful, as it should be a full 12, as 6 double bonds with 2 hydrogens being added to each should give 12. However, this is only given 6, allowing us to believe that this mechanism is flawed. Further information for the mechanism not working well and being flawed is that in a reaction of N2H with only nitrogen gas and regular glacial acetic acid, we were still able to successfully synthesize stercobilin, which, based on the current drawn-out mechanisms for hydrogenation, should not be possible at all, as only nitrogen gas would be added across it, which is not theoretically possible, indicating that the solvent itself is protonating across those double bonds, reducing the molecule to bilirubin.
So this semester, to better investigate the mechanism of hydrogenation, we decided to choose 2 new molecules that were kind of similar to molecules in bilirubin. So the 2 that we chose were limonene and alpha-flandrine. And we picked these two just because when you hydrogenate both of them, they end up giving you the same product of p-methane, which is shown on the right. So this is just a reference slide for the IR of p-methane. So this is the product, what we should be getting when we hydrogenate both limonene and alpha-phenandrine. And then this is the reference, again, what we are looking for when we analyze our products for limonene and p-methane, or limonene and alpha-phenandrine. So when determining like the reaction length we wanted to use for the hydrogenation mechanisms, we first ran the H2H of limonene for 15 minutes. And after we did that, we noticed a small peak between 5 and 6, which is circled on the left image. And so we decided to run the reaction for an hour just to see if we could get rid of that peak. And obviously we did that on the right. You can see that the peak is gone between 5 and 6. And what we determined was that with the 15 minutes, we were able to break the bond on the outside of the ring of limonene, but the ring or the bond on the inside of the ring was still there after 15 minutes. So with that being said, we determined that we would do the reaction for 1 hour. So this is our H2H IR of limonene, and here you can see that it looks just like the reference of p-methane and showing that we had all single bonds. And this is our H2D of limonene. We have the IR and the NMR. And if you look at the NMR on the right, you can see the taller peak at 7. And this is actually our solvent peak. And that is the one difference between looking at the reference slide of p-methane. Other than that, we were able to hydrogenate limonene doing the H2D reaction. And then we performed the D2H of limonene only because we actually ran out of the deuterium gas. But here again, we're able to see that we could completely hydrogenate the limonene molecule to p-methane. And here we have the H2H of the alpha-phenandrine molecule. And again, we have our solvent peak on the NMR at 7, and we were able to get rid of all the double bonds, and we are left with all single bonds in the p-methane molecule.
So some conclusions is that D2H of bilirubin was not able to fully label stercobilin with the 12 suggested deuterium atoms. And that leads us to believe that the mechanism for hydrogenation may be wrong, which will, for a fact, hinder our research further into stercobilin, as if we cannot fully understand the mechanism, then we will not be able to run successful reactions over and over again in order to make this isotopically labeled biomarker for autism. We also found that without hydrogen gas present, stercobilin could still be made in an N2H reaction condition, further showing that the reaction mechanism is not correct. We also are able to fully successfully hydrogenate limonene and alpha-phenandrine. Currently, because the semester is cut short due to the COVID-19 crisis, we are not able to finish total data collection of the reaction conditions we wish to do for limonene and alpha-phenandrine. But in the future, we'll be changing the reaction conditions such as the pressure, the time, and the temperature, as well as solvents and gas used in order to try and find out if double bonds are reduced and how they're done. We'd also like to give a special thanks to the Nazareth College Department of Chemistry and Biochemistry for all of their support throughout our research, the University of Buffalo for allowing us to use their instrumentation as well as their assistance in our project, Dr. Amber from Charlebois for her guidance as our research advisor, Mrs. Jane Schieber for her assistance in all our laboratory needs that we need. She's the best in Peckham Hall. And also we'd like to thank all previous undergraduate students who have worked on this project. Thank you for your time. And if you have any questions, feel free to contact us.